Hi, I'm Natasha and I will be discussing parts of my PhD on the maternal and infant gut microbiome in fetal alcohol spectrum disorder. Fetal alcohol spectrum disorder is an overarching term describing 4 diagnoses along a spectrum of increasing severity consequential to prenatal alcohol exposure. Despite the fact that prenatal alcohol exposure is one of the most preventable causes of birth defects, developmental disorders, and mental retardation, South Africa has the highest prevalence of FASD worldwide, with the Western Cape prevalence reported to be between 20 and 28%, significantly higher than the global prevalence of 0.77%. The gut microbiome, which represents the microbes and their genes in the human gut, plays a role in regulating nutrient intake, intestinal motility, neuroimmunity, and metabolic and immunological development. Ongoing research suggests that the gut microbiome may play a role in mental health via the gut-microbiota-brain axis, the bidirectional communication axis between the gut microbiome, central nervous system, and gastrointestinal tract. Changes in microbial composition and functions from their normal beneficial state to a state that is deleterious to the host's health may have detrimental consequences for brain, metabolic, and digestive disorders, influencing the risk of developing behavioral changes and neurodevelopmental and mood disorders. Excessive alcohol intake has been found to affect gut microbial composition. An altered maternal gut microbiome may translate into abnormal infant gut colonization, thereby resulting in altered infant gut microbiota functioning. Initial gut bacterial colonization plays a fundamental role in brain and neurodevelopment, both pre- and postnatally. An infant's evolving cognitive and neural development is dependent on microbial composition and the corresponding microbial metabolic outputs. The study therefore aimed to compare the gut microbial composition of women who consumed alcohol during pregnancy and those who abstained from drinking alcohol throughout pregnancy. The study also aimed to compare the gut microbial composition of infants who were exposed to alcohol prenatally and those who were not exposed to alcohol prenatally. Pregnant women were recruited from antenatal clinics in Robertson and Wellington. The audit test was used to assess alcohol use during pregnancy. Focusing on past month drinking, an audit score of 8 and above indicates hazardous drinking. An audit score of between 4 and 7 indicates moderate drinking, and an audit score of 0 indicates no drinking. A woman was assigned to the case group if she had an audit score greater than 4, while a woman was assigned to the control group if she had an audit score of less than 4. An infant was assigned to the prenatal alcohol exposure group if its mother had an audit score greater than 4, while an infant was assigned to the no prenatal alcohol exposure group if its mother had an audit score of less than 4. Stool samples were collected from both the mothers and their infants, and microbial DNA was extracted from these samples. The microbial DNA was subjected to paired-end sequencing of the V1 to V2 region of the 16S ribosomal RNA gene. The Data2 pipeline was used to pre-process the FASD sequencing files, construct an amplicon sequence variant table, and to assign taxonomy using the Ribosomal Database Project Reference Database. PhyloSeq and R were used to rarefy the data to correct for differential sequencing, perform analyses to identify whether there were compositional differences between cases and controls, calculate alpha and beta diversity, and compute the statistical analyses of microbial composition. To date, 16S sequencing data from only 77 pregnant women has been released. The results discussed hereafter are based on the microbial data from these pregnant women. The gut microbiome of both the cases and controls was dominated by Prevotella in purple, Bacteroides in orange, and Succinovibrio in pink. Prevotella and Succinovibrio are associated with plant-rich, high-fiber diets, while Bacteroides has been linked to a high intake of fat and protein. There were no significant differences in the relative abundance of taxonomic units between cases and controls. None of the variables investigated, namely age, pregnancy-adjusted BMI, trimester, or Bristol stool scale, were associated with the relative abundance of a single genera or group of genera. For microbiome analyses, we commonly look at two different types of diversity. Alpha diversity represents the diversity within a single sample, while beta diversity represents the diversity between samples. There were no statistically significant differences in alpha diversity between cases and controls. There were also no statistically significant differences in beta diversity measures between cases and controls. Of the variables investigated, trimester may have an effect on Bray-Curtis distance with an adjusted p-value of 0.096. Major alterations in gut microbial composition occur during the third trimester of pregnancy. Estrogen and progesterone impact the composition of the gut microbiome through their effect on bacterial metabolism and result in an increased abundance of the phyla Proteobacteria and Actinobacteria and a decrease in the genus Faecilibacterium. Although no significant differences were identified, differences may become evident with the larger sample size. An altered maternal gut microbiome may influence initial bacterial colonization of the neonate gut. Abnormal neonate gut colonization may alter the functioning of the infant's gut microbiota, which may have significant long-term health consequences. I would like to thank my funders, and thank you for listening.